Thank you for the introduction, and I'm really delighted that I'm talking after Gansi gave a really amazing introduction to my work. So without further ado, let's start. Next 10 minutes, I will be talking about the application also for Nanopore in unraveling complex Mendelian cases. I will be discussing specifically the GREGoR project, which stands for the genomic research for to elucidate the genetic of rare diseases. On this project, we are aiming to connect the relation between, or the linking the disease to their genetic causes, which actually allowing us to improve genetic testing by identifying which genotype is associated with this phenotype. Doing this so shortening the time for diagnosis between the onset of a disease and the actual diagnosis. Currently, there are 5 research centers that are working on this project and 1 data coordinate center. Additionally, in this project, we are focusing basically on the Mendelian cases and rare diseases. By rare diseases, I mean any disease that are affecting around 200,000 of American citizens, and roughly there are 7,000 rare diseases affecting 30 million Americans. So now it's a time to ask actually why we need such an initiative. What is the important role? What we are missing so far? To answer this question, actually, there are different points that I need to highlight. First of all, while exome sequencing is highly crucial and managed to actually resolve a lot of diseases by identifying a variation in protein coding region, but this does not come without any flaws. First of all, we are only focusing on 1 to 2% of the entire genome. We could have some region of poor capture region. Additionally, we are overlooking some important non-coding region and regulatory element which play a crucial role in diseases. Add to that, actually, exome sequencing is ineffective in complex genetic condition where there are multiple genes are interplaying together to show the phenotype. Besides that, we are missing the structural variant out of the exome region. Also, we are not able to identify SNVs in complex medical region. We are missing methylation and also phasing. Finally, the most important point actually is that whole genome sequencing actually is now cost effective. Based on this information, we actually published a paper to show exactly what we are missing. So far, I think now you know what I am doing and actually what is my role in this project to solve the unsolved diseases. But how many diseases actually I'm talking about? In the project here, we have more than 1,600 individuals that are enrolled. More than 400 of those are families. If we look to figure A, 76 or more of these cases are entry, which is really important, so we can compare cases to each other. We are actually here covering different range of diseases or disorder, around 284. If we take a look to figure B, around 60% of those are neurological diseases, and followed by 10% which cancer diseases, but we have other range of diseases that we are looking to. Add to that, as you can see in figure C, we are actually also showcases in different ethnicity, from Native American to Caucasian and other ethnicity, which is really crucial and important if we need to build a database, as mentioned by Gus, for different ethnicity and how a structural variant could affect, or actually variant in general, not only structural variant, could affect people from different backgrounds. To solve or actually to answer the problems that I showed in the previous slides for exome sequencing, we need whole genome sequencing, we need long read with high accuracy, we need to identify structural variant accurately, we need to identify SNVs in complex region, and phasing. If you are here now, I would say you understand what I will mention next, so also for Nanopore, to give us the opportunity to answer this question that I raised a second ago. So let's move to the number of cases that I'm showing today. I'm focusing today on the pilot data, where we are actually focusing on 40 samples, 3 of those are families, 12 families and 3 singletons. These data are really old, so you don't expect the coverage to be really high because of the age of the sample. But however, we have a good coverage between 15 to 35 with enough read less than N50 to span the stretches of repeats and identify structural variant accurately. So the next question, how I will answer or how I will analyze this data. Previously, I developed the process, which is a comprehensive framework for calling genetic and epigenetic variation, and also it phases them. Have tons of tools or state-of-the-art tools that works in a cohort mode or in really dynamic mode to identify all types of variant and phase them together. It seamlessly installs through Conda, so you do not need to focus much on installation. Additionally, we can identify the required parameter for each tool to make it simple and clear and run it easily. We have a really number of results that we got from process, ranging from QC for each step, calling variant and phasing them. This is a big project. If you need to talk more about it, you can talk to me later. So this is the first part of the result. I'm showing here only 3 samples. What I'm showing here is analyzing of 3 UBs. If you look to figure A, which actually represents SNVs between proband and the parent, we are comparing head-to-head to identify the novo variant. So we can look to them first before moving forward to other heterozygous variants that are in the proband and absent from one of the parent. As you can see, around 40% are shared, and 4% of those are unique or de novo for the proband, which we'll be focusing on the next slide. Looking to the structural variant, here we have less than 1% unique to the proband, which roughly around 200 structural variant, different from sample to sample. The next question, we have a really high number of data here, and SNVs and indels structural variants, so how we can effectively analyze them. Here I propose an annotation pipeline for SNVs and SNVs that take advantage of what we have already, like database. For SNVs, we start by filtering by a need frequency in the population, and then we are annotating them based on coordinate or functional impact, plus other factor like Dana score, which stands for the literate annotation of variation using neural network, which is somehow a way of periodizing your variant. For structural variant, we have different tools, like tool depend on the population structural variant, annotating them like 1000 genome and genome. However, as I just mentioned a second ago, this was done by short reads, so not all of the information available from the database will be beneficial to my case. And plus, of course, the gene names that could be crucial for some cases. One of the issues that I just mentioned, that when even I said there are 1% or less than 1% of the structural variant unique to proband, 93% of those I cannot annotate because of the lack of information. Thus, we need to actually identify structural variant more accurately in larger project to be able to have a better understanding by expanding our base knowledge of databases or structural variant and their relation to diseases. That's why I actually propose the approaches that I'm developing now, which could be a cornerstone for further analysis in cohort to look to the structural variant SNVs and indels comprehensively and build such a kind of database which will actually enhance our understanding of diseases and make us actually reduce the time we need to pinpoint cognitive variation. Initiatives like All of Us and 1000 are actually working on that. So for case of time, I will only show one case that we are focusing on. It's for a 10 weeks old male with a CHARGE syndrome, have a developmental disorder and several defects. They already managed to do exome sequencing. Sadly, it was inconclusive. There are more than 500 genes that could potentially be causative for this case. So, as you know now, we have identified a really large number of SNVs and SNVs focusing or actually affecting this set of genes. Applying our Brensis tool plus the annotation pipeline, we managed to classify them or stratify them into tiers and look to them one by one. So we have actually reduced the time in identifying the case. Here I am showing an IGV screenshot. The blue bar represents a structural variant. It's around 2,474 base pair that affecting this. You have 3 panels, the son, mother, and father. Any gray line represents a read, and any dark line represents a deletion. As we can see here, we have a heterozygous structural variant that affecting the son, absent from the mother, and there are 8 reads, if you can see, in the father. However, we could not identify structural variant in the father, which suggests it could be a mosaic variant. Thus, we have some kind of skepticism if it's causative or not, meaning we need to do more research to prove that this heterozygous structural variant could be associated with this disease. Specifically, it's affecting the first exon of 2A3 gene. Beside that, I just want to conclude because I don't have much time left. So, first of all, we managed to identify a coronary resource analysis pipeline that I show you. Actually, they are moving head-to-head with this annotations pipeline. And we will extend it more to include shorter tandem B and copy number variation, so we could answer more questions. But what I really interested in, it sees that we need to build a more robust strategy for sample analysis and stratifying of analysis, how we could do that and build a role model. What I'm suggesting here could be applied for different scenarios, not only families. So we need to move from a single family to cohort level to have a diverse database and better understanding. And this could be a role model actually for more extended research. So lastly, the most thing or the result from all of that will be improving our annotation first, which will lead finally to the ultimate goals that I'm aiming to integrate long read to be an initial step in clinical setting and paving the way for personalized medicine. With that said, I think I exceeded the time already. Okay, sorry. So I would like to thank you all for your patience, and I'd like to thank the College of Medicine Human Genome Sequencing Center and GREGoR Initiative and all my colleagues. Thanks once again for your patience and for your attention. Thank you.